The measure of intelligence is the ability to change.
Hi all, Dr. Clark here, and today we're going to talk about anaerobic metabolism. So we left off, we were talking about the Krebs cycle last time, and so that's pictured here. Here's a mitochondria, and here's that Krebs cycle. We also talked about the electron transport chain or electron transport system. Okay. Prior to that, we talked about glycolysis over here in the cytosol or cytoplasm of the cell. Now, we're going to try to put it all together. Remember that glycolysis is the breaking of a glucose molecule into pyruvate. That generates 2 ATP. That pyruvate can be processed in two ways. It can either be processed with the presence of oxygen inside the mitochondria, and that process is aerobic metabolism, and that's going to generate 34, 36 ATP. Or it can be processed without oxygen through anaerobic respiration, through the process of fermentation, and either form alcohol or lactic acid. Okay, so we're going to cover these steps and kind of this part here, this anaerobic respiration and what's going on. So when there's no oxygen present, the organism generates very little ATP. So with the development of oxygen in the atmosphere, so when plants came and they started kicking off oxygen as this waste product, and oxygen started, being part of the atmosphere, life on Earth just exploded. Because now you can take that glucose molecule, instead of getting 2 ATP, maybe a little bit more ATP out of that single glucose molecule, now you're getting 36 ATP because oxygen's present. So you have this capability of, really expanding life. Right? Before, energy was the limited resource, and now you have a better way to process that energy and get more out of that original food molecule to drive work. So organisms could get bigger. They could get more, have different structures. They could get more complex because of the presence of oxygen. Now, without oxygen, there are ways at which this material gets processed. It doesn't just stop. Glucose molecule gets converted into pyruvate. It doesn't just stop at pyruvate. Okay, if there's no oxygen, it has to be further processed, and it does so in a couple ways. That further processing is called fermentation, and it's called fermentation in lots of different organisms. There are different ways at which fermentation can occur. And the process of fermentation is taking some of those electron carriers, electron carrying molecules, taking electrons and hydrogen ions from them. Okay, so remember, in glycolysis, they were generated. So glycolysis generates two main products, a little bit of ATP, and electron carrier. And then it's not really a product, but it's the end result of glycolysis is pyruvate. Okay, so you could think of it as three main products. That pyruvate, if there's oxygen, that's definitely a product because that's going to be further processed for energy perspective. Okay, if the pyruvate is not in the presence of oxygen, then you got to deal with that pyruvate in a different way. Okay, and that's exactly what fermentation is. Okay, it's taking the hydrogen atoms and the electrons from that electron carrier that was generated in glycolysis. It's taking that and it's donating those to the pyruvate. Right? So it's donating that material to the pyruvate to further process it. Right? That NAD plus, which just lost its hydrogen ion, is now regenerated. It can go back into glycolysis. Okay? Another glucose molecule can be broken down. Another hydrogen ion and electron can be donated to the NAD plus, and it can be regenerated. Okay? Now, we're not going to dive into prokaryotes when it comes to this process. I just want you to note that prokaryotes have numerous ways to go about fermentation. In fact, I'm 99% sure that when we discover more prokaryotes, we'll probably discover other ways at which they can do fermentation. This shouldn't be a big surprise. They've been on the planet for, probably 2 billion years, probably maybe even more than that, 3 billion years, and they've been doing glycolysis this kind of whole time. They've been breaking down glucose molecules without oxygen. There's lots and lots of bacteria that occur in regions that are void of oxygen. And they've been breaking down glucose molecules into pyruvate and then taking that pyruvate and processing it further without oxygen. And so there's lots of different ways that they can do this. We're not going to dive into them. What we are going to talk about is the eukaryotic ways. Now, there might be even a few eukaryotic ways that haven't been discovered or really worked out the logistics, but there are two main ways at which eukaryotic cells, eukaryotic organisms go about fermentation. The first one is fermentation that you're probably really familiar with because lots of college students, lots of people out there that are above the age of 21 and sometimes even below the age of 21 consume the material of this fermentation process. Okay, and that would be ethanol or alcohol. Okay, so organisms like yeast, like single-cell fungi, plants can do this. There's lots of organisms that can do fermentation in this way and produce ethanol. Okay? It is taking that pyruvate and utilizing the energy from NADH, the hydrogen and the electron, to further process acetaldehyde. Okay? Acetaldehyde is a molecule that can be further processed to form ethanol or alcohol. Okay? The other way to do this, and this way is done in animals, and that is to take that pyruvate and convert it into lactate or lactic acid. Okay? And so two main ways, either the plant, fungi, some protista way, formation of ethanol, or animal and some protista way, the formate of lactate or lactate acid. Okay, so here you can see the two processes. Ethanol fermentation. You're taking that glucose, you're going through glycolysis, generating pyruvate. Remember that process of glycolysis, you're generating ATP, a net gain of ATP, 2 ATP. You're also generating NADH molecules, these electron carriers. That pyruvate can be processed further into acetaldehyde. Right? Carbon dioxide is removed. Acetaldehyde can be further processed into ethanol. Lactic acid, same general process, very similar. Okay? ATP generated, NADH2 generated from glycolysis, pyruvate, but now the pyruvate is just processed directly into lactate. Okay? Now, this lactate or lactic acid Many of you felt the production of this. Okay, so if you were to just stand up and go run 100 yards, maybe 200 yards, it just depends on how often you run. It could take a lot more than that. But if you were to run without stretching, okay, so just muscles are have plenty of glycogen, they have plenty of sugars in them, they haven't been used readily right away, kind of void of oxygen. And if you were to just go run as fast as you can, okay, what's going to happen is you're going to go through this process. Glucose, okay, is going to go through glycolysis. It's going to generate some energy. It's going to generate some electron carriers. It's going to generate that pyruvate. That pyruvate then, because there's no oxygen around, it's not going into your mitochondria. That pyruvate is going to be processed via that NADH, those electrons being donated to it to form lactic acid. And your legs will burn. Okay? That burning feeling is coming from that production. Okay? That anaerobic production of lactic acid and the anaerobic kind of process of breaking down that glucose molecule. Okay, so we already talked about this a little bit before, but I just want to reiterate that glucose, carbohydrates are not the only food source, of course, and it's not the only pathway to get energy. So when we talk about carbohydrates, we know that glucose is a monomer. Glucose is this building block for complex carbohydrates. So if you bind a bunch of glucose together, you get starch. If you bind a bunch of glucose together in different ways with a few different bonds, you get cellulose. Right? So there are glucose molecules or building blocks that are built of glucose. Well, first you have to break those molecules down. You have to break that starch down. You have to break that sucrose down to get to glucose. Okay? And then it can go through glycolysis and it can go through that pathway. Okay? So just, as a recap to that, any complex molecule is going to be first digested into simpler subunits. Okay, so this is taking polymers, complex molecules, and digesting them into monomers. So if you have a disaccharide, you're going to digest it into a monosaccharide. Okay? Now, these processes often take numerous enzymes, and these pathways can be just as complex as the other pathways like glycolysis and the Krebs cycle. Okay? When that process gets that intermediate, then that intermediate can enter the systems that we've talked about, the cellular respiration systems we've talked about. Okay? And like I said before, intermediates, different types of intermediates, enter the system in different places. Okay? And we're going to kind of look at some of those examples now. So remember back, quite a few lectures ago, we were talking about biomolecules. And I said there are 4 biomolecules: nucleic acids, proteins, carbohydrates or polysaccharides, and lipids. So those are your 4 biomolecules. Each one of those is a polymer. Each one of those can be broken down into its building blocks or its monomer. So nucleotides build up nucleic acids. Amino acids bound together with peptide bonds make proteins. Sugars such as glucose and fructose make bigger sugars, polysaccharides. Fatty acids bound with glycerol, etc., will make lipids. Okay? So these are the building blocks. Now these building blocks can even be further broken down to enter into this system. Remember, the system that we're interested in is glycolysis, or the one that we just covered, pyruvate processing, the Krebs cycle, and then the electron transport system. Okay, so if you're talking about nucleotides, nucleotides can be converted into amino acids. They could be converted right into a product inside the Krebs cycle, or they can just be converted into ammonia. Ammonia. Okay? Amino acids, same thing. And these amino acids, ammonia, these amino acids can be converted into pyruvate. They can go directly into the Krebs cycle. They can be converted into nucleotides. There are lots of pathways for the amino acids that are being broken down from the proteins. Sugars, pretty much one way, the way that we just talked about. This is what makes carbohydrates an excellent source of energy, is those carbohydrates have a fairly streamlined system. Right? They're not bouncing around. They don't have the ability to enter the system in at many different steps. Okay, they're entering in and they're following a streamlined linear system. Lipids and fats, fatty acids, okay, they're normally just converted into acetyl-CoA. Now, remember back when I was talking, if the system has already got too much ATP, these intermediates that are coming in, it doesn't matter what intermediates are coming in, they're going to be converted into acetyl-CoA. Okay, except for when we talked about nucleotides, I told you, nucleotides are not really good for energy sources. Nucleotides are good for maybe trace elements. They're good for information holding molecules. They're good for building blocks for amino acids. Okay, nucleotides, but they're not really good for as an energy source. Okay? But these other three are excellent energy sources. And you can see they all have the ability to be converted into acetyl-CoA. If you have too much energy in the system, acetyl-CoA is going to turn into fat. Okay, it's going to go through the process of generating fat. If you have not enough ATP, then it's going into the Krebs cycle. Okay, so hopefully that makes sense. And we're going to progress now, and we're going to start looking at genetics and DNA and some other components to that. So then we can start talking about gene therapy, and we can start talking about cancer treatments and CRISPR, some of the other issues that occur in today's society that are very important. Next time.